In this video, let us see about the double fertilization, the endosperm and the embryo. Come, let's go to the video. First, let us see what is double fertilization. So in angiosperms, there is a process called double fertilization. You know very well that in pollen grain, there will be formation of two male gametes. Generative cell will divide and give rise to two male gametes, where one male gamete will fuse with the egg. So one male gamete fuses with the egg. That is the male nuclei fuses with the egg. That is called syngamy. That is called fertilization. And two, another male gamete fuses with the polar nuclei. Or the secondary nucleus. So one male nuclei fuses with the egg cell, that is called fertilization of syngamy. Another male gamete or male nuclei fuses with the polar nuclei, which is now known as secondary nucleus. So this is called double fertilization. One male gamete fuses with the egg cell, the other male gamete fuses with the polar nuclei or secondary nucleus. Okay, here one more concept is hidden, that is called triple fusion. Okay, triple fusion in the sense in the embryo sac there are two polar nuclei. These two polar nuclei will join to form secondary nucleus. To that secondary nucleus, another male nuclei get fused. As there is involvement of three nuclei, it's called triple fusion. As there is involvement of three nuclei, it's called triple fusion. Three nuclei in the sense two polar nuclei. To that one male nuclei. So 2 plus 1, 3. Then it becomes a triploid. So this secondary nucleus, which is triploid in nature, will give rise to this is the endosperm. This secondary nucleus, which is triploid in condition, will give rise to endosperm. That endosperm is triploid in nature. Okay, if you see that embryo sac diagrammatically, it will be like this. So this is the egg cell. Okay, this is polar nuclei. These are the antipodals. Okay. This is the cell end. This is polar nuclei. This is egg cell. These are synergids. So that's what I told. One male nuclei will fuse with the egg cell. Another male nuclei will fuse with this polar nuclei. Before it fuses, this will join. To that, another male gamete joins here. So this is called triple fusion. As there are two male nuclei involved in this fusion, it's called double fertilization, right? Now let us see about endosperm. So endosperm Actually, the primary endosperm cell. Okay, it divides and gives rise to, as I told you, triploid endosperm tissue. The primary endosperm cell divides and gives rise to triploid endosperm tissue. Okay, and it has reserve food material. So it is rich in reserve food material. So it's in reserve food material. Okay, because it has to provide nourishment to the developing embryo. This is having the food content. Okay. Similarly, the primary endosperm nucleus. Okay, shortly known as PEN. It undergoes successive divisions. Okay, to form free nuclear endosperm. Okay, as many as nuclei will be there, it will be free nuclear endosperm. That later on will become cellular endosperm. So initially free nuclear endosperm without any cross walls. Later on cross walls will be formed and it will become cellular endosperm. Right. Now I'll give example for this free nuclear endosperm. For free nuclear endosperm, that coconut water that you take from a tender coconut, that's nothing but free nuclear endosperm. Okay. So coconut water from tender coconut. That's an example for free nuclear endosperm. And for cellular endosperm, the same coconut has that white kernel. That white kernel is called the cellular endosperm. Right? These are all the examples. And in some cases, the endosperm is fully consumed by the developing embryo. In some cases, the endosperm is fully consumed by the developing embryo. For example, in pea, groundnut, and beans. Whereas in some cases, that the endosperm will persist till seed formation. For example, castor, coconut. Okay. Okay, endosperm fully consumed. Example, pea, groundnut, bean. Here, endosperm persist till seed formation. Example, castor, coconut. Now we'll see something about the embryo. Okay. So usually the embryo develops in the micropylar end. Develops in micropylar end. Embryo develops in the micropylar end. Okay. Then the developmental stages of embryo is called embryo journey. The developmental stages of embryo is called embryo journey. So the developmental stages of embryo is called embryo journey. Embryo develops from the micropylar end. Okay. So actually this happens like this. Zygote, you know, the product of fertilization, it will give rise to pro-embryo. This pro-embryo will modify into a glandular embryo. This glandular embryo will become a heart-shaped embryo. And finally it becomes a mature embryo. So these developmental stages are called embryo journey. Yes. Now we'll see how that embryo is there in dicotyledons and monocotyledons. Okay. In dicotyledons, as the name tells you, it has two cotyledons. Cotyledons means seed leaves. Okay. These seed leaves will be present till true leaves are formed. Once true leaves are formed, they will undergo photosynthesis. Okay, so till then these cotyledons will support the plant. Okay, cotyledons are called seed leaves. So in case of dicotyledons, it has an embryonal axis. So it will be like this. So this center portion is called the embryonal axis. Okay, this is cotyledon. Okay, here also this is one cotyledon, this one cotyledon, two seed leaves. Cotyledon. This is the radicle, that is baby root. Okay, this one is the plumule, that is baby shoot. So the center part is the embryonal axis, okay, and this is the budding root. It's called the baby root. The upper portion is the baby shoot called plumule. This is the embryonal axis. It has two cotyledons. Cotyledons are seed leaves. Cotyledons will be very thick because of the reserve food material. So as the new leaves start growing, as the plants start growing, all the food material from these cotyledons will be used up. Later on, the cotyledons will dry and they will fall off. Afterwards, this plumule will become, and it will just grow and the leaves will start doing the photosynthesis. And two more definitions. So the portion below the cotyledons is called hypocotyle. Hypo means below. The portion below the cotyledon is called hypocotyle, and the portion above is called epicotyle. The portion above the cotyledon is called epicotyle, that which is below is called hypocotyle. So now let's see how it is happening in monocotyledons. Okay, just I'll show the structure of the cotyledon here. Okay. So it will be like this. Okay. So this is the seed of monocotyledon. This is called the pericarp, the outer covering. This will be the endosperm, the nutritive tissue. Okay. And actually this is the cotyledon called scutellum. Here the cotyledon is called scutellum. Okay. This one is called coleoptile. Okay, and here it is called coleorhiza. So it's a sheath, the membranous sheath that covers the plumule is called coleoptile, and similarly a sheath that covers the radicle is called the coleorhiza. Coleoptile, coleorhiza. Coleoptile sheath that covers plumule, coleorhiza sheath that covers the radicle. So of course this is the plumule and this is the radicle. So it's a single cotyledon, can see the pericarp, endosperm, the cotyledon, exact cotyledon is called the scutellum, and this plumule and radicle are protected by the sheath by name coleoptile and coleorhiza. So in this video, we learnt about the double fertilization, triple fusion, about the endosperm tissue and the embryo, the cotyledons. So with that, we'll wind up today's session. Hope you enjoyed this video. Kindly like, share and subscribe. Thank you.